Hello, HL seniors. Welcome to an abbreviated version of Topic A, Section 1, Neural Development. So this is our first video lecture. So what I'm shooting for here is sort of a shorthand version of the section. If you find that you are not satisfied and you want some more detail, feel free to read Section A.1. I'm also pulling from the Topic 6 0.5 material, which is on neurons and synapses. So if you feel like you need a little review, go ahead and reread or reflect on 6.5 and 8.1 as needed. Of course, this is not strictly following the syllabus because you're not taking your exams, so we will wander in and out of the detailed syllabus structure as needed. We'll skip some of the nitty-gritty concepts and focus on the big picture here. All right, here we go. So, to begin, what are neurons? Neurons are sometimes referred to as nerve cells. These are the cells in the nervous system, and they're designed to carry electrochemical impulses in one direction, from the cell body, or the soma, toward the terminals of the axon. So let's talk a bit about the structure of your typical neuron. So this is sort of the input of a nerve cell, if you will. This is where this nerve cell is going to receive stimuli. Then, if it's stimulated enough, it will send a signal down its axon to communicate with other cells. Now, these cells could be more neurons, they could be muscle cells or glands. In any case, they're going to have to communicate with other cells to perform a function. All the nerve cells are doing is relaying information, telling cells to activate or perhaps inactivate, depending on the situation. So this nerve cell is going to be initiated or activated. And the way this works is that as this nerve cell is activated, it's going to change its electrochemical potential, which sounds complicated for those of you outside of physics or chemistry, but the idea basically is that it's going to pump ions, positively charged ions, in and out of this cell. As it does that, it changes the charge of the cell. And when that charge changes, it can carry a flow of the charge down the cell toward the end down here. This requires a lot of active transport of ions, and this is why your brain requires so much energy, because you need lots of ATP to power that pumping of ions in and out of the cell to maintain the proper charge to carry that signal. If we were to look at the graph of the charges, we're looking at what's called the action potential of your neuron. When this nerve cell is resting, it's here. It has a negative charge, negative potential charge like this. Then, as this nerve cell receives some stimulus at the dendrites, it has to basically receive enough of stimulus, enough stimulation to start to activate itself. This is what's called the threshold. So, a really twisted example is this typical physiology lab where you dissect a frog, you steal its muscle, and then you give that muscle an electrical impulse. If you give it a very low shock, it'll just, maybe bump up a little bit, but it won't reach enough potential, if you will, to cause the muscle to actually contract. It's only when the electrochemical potential is reached that this nerve cell will actually fire. This is called the threshold potential. So nerves work on an all or none principle. Either you hit the threshold and the action potential occurs, or it doesn't. And so sometimes you can get to a point where your nerves become so stimulated they go into a state of hyperpolarization, and this leads to basically a lack of sensitivity. You've probably experienced this where you can hypersensitize or become numb to a certain sensation where you no longer feel it because those nerves have been used so many times they can't get back up to that threshold potential to function again. This can cause muscles to twitch rather than coordinate their movements, or you could lose sensation in your feeling or something. You can also tune out a sense of smell if those neurons become hyperstimulated. Now, getting back to the main big picture here, we're going to focus today on how these nerve cells communicate with each other and how they originate in the first place. So, the way this works is that a nerve cell or a neuron is going to communicate with another neuron. And they do this by sending a message down the axon and then linking up with the dendrites of the next cell. whenever they form these little junctions or these communication points, there's a little bridge where the cells don't actually make physical contact, but they come quite close to each other at a region called a synapse. This is a blown up picture of a synapse. All of these little connecting points and connecting points over here, these are all synapses. Here's a close-up view of a synapse, a junction between cells in the nervous system. Here's our presynaptic cell. If its action potential is reached, then it will communicate with the postsynaptic cell. Here's another view of a synapse. And so in this case, our presynaptic cell has reached its action potential, and when it does that, it's going to release these little chemicals, which are called neurotransmitters. So once this cell is activated, it releases its neurotransmitter. These neurotransmitters will then migrate across here to our postsynaptic cell, and they're going to bind to a receptor. And now our postsynaptic cell is going to be activated. So now we have a stimulus, and if that stimulus is strong enough to hit the threshold potential, now our second neuron is going to fire. And so now these two nerve cells, these two neurons, have successfully communicated with each other. This is called neurotransmission. It's a process of cells communicating with each other. Nerve cells specifically communicating with each other. This all occurs at a synapse. Now, synapses can occur between two neurons. They could occur between a receptor cell, something that senses a sensation like a smell or some kind of touch, and then they could synapse between a receptor and an effector cell, causing a response to a stimulus. Or they could connect to a muscle or gland and cause very specific metabolic activity. In any case, these synapses are important points where these cells communicate with each other. And today we're going to focus on how these synapses form and where the neurons come from in the first place. And it all begins with embryological development. And so here we have the fertilization of an egg, where the sperm nucleus and the egg nucleus fuse to form a fertilized zygote, which then divides into undifferentiated embryonic stem cells. This is a blastula. And then the next step is that these cells are going to start to differentiate just slightly. They're going to start to form very specific layers, which make up the whole makeup of your body plan. So the outer layers of your body, picture your skin, some attached muscles, would be the ectoderm. In between the mesoderm, and then the inside is the endoderm. So these three layers start to form different organs in your body. Today we're just going to focus on how these three layers start to roll around and fold into your central nervous system, your brain and your spinal cord. So we're going to be focusing on this layer right here. Now on this next slide you can see how those three layers start to fold over. Again, I'm going to back up. This is the region right here. And imagine this fold occurring right at the top. Now this folding process is the very beginning of your entire nervous system. This process is called neurulation, where the ectoderm, folds over and starts to form what's called the neural tube. This is what happens. Here's that ectoderm layer up here. It folds around, forming this large ridge. Oops. That ridge then continues to fold over and then fuses, and then the inner layer forms a tube, which looks somewhat like this, and this is what becomes your spinal cord. Now this tube starts to differentiate into the more posterior region, which becomes the brain, and the more anterior region, which becomes the brain, and the more posterior region, which becomes the spinal cord. So the front becomes the brain, the back becomes the spinal cord. Now this folding over process forms the neural tube, the spinal cord. It also forms some protective layers around that spinal cord. And imagine if this process does not happen properly, you'd be born with your spinal cord on the outside of your body. It's quite dangerous. It's a condition called spina bifida. It looks somewhat like this. And so this infant was born with that neural fold and neural tube forming improperly. Again, this is called spina bifida. We can now fix this with surgery, where you can essentially just push this back in and close in this gap where there should be protective coating and skin. Obviously, it's quite dangerous to have your spinal cord reaching the outside of your body. We can just pack that in and protect the spinal cord from damage. So once this neural tube has formed, again, it differentiates and forms the basis of your central nervous system. What happens is these neurons, they start to change, and these axons will grow out into different directions, and they'll form synapses, points where they can communicate with the rest of your body. And so this is a very strange looking diagram, but this is basically what happens as this differentiation occurs. Down here in yellow, this is where your spinal cord continues. All up in here, this is where the brain stem starts to form. Cerebellum and cerebrum will start to form in this region. And so this neural tube starts to differentiate to the central nervous system. That's your brain and your spinal cord. Now, as this occurs, you start to form more and more synapses at all of the axons and all the dendrites from all the neurons in your central nervous system. In fact, you're going to form far more synapses than you will ever use. The body does this intentionally. So when you're born, you have a certain number of neurons, which then will continue to synapse with other neurons. They form more and more dendritic and axonal connections between these nerve cells. As this occurs, you start to form memories, and you start to build skills, if you will. So imagine one of these connecting points is you at 6 years old trying to learn how to play the flute. Now, as you grow older, you realize that you hate the flute, you no longer practice your flute, you give up on your flute lessons, and what happens is you're going to stop using those flute memories or those flute connections in these synapses, and then you're going to cut it away. You're not using it, and so you get rid of it. This is the process of neural pruning, where you start to cut away or remove the connections that you're no longer using. And of course, neural pruning sounds like it could be a problem. You're losing memories, but in fact, it makes the brain much, much more efficient at performing the functions that it needs. So at 6 years old, you have a great deal of what's called neuroplasticity, where you're able to learn all kinds of new activities. And then as you age, you start to get rid of some of those connections, those unnecessary axons. Yes, and also some of those unnecessary synapses or connecting points between nerve cells. This whole process of getting rid of those connections is neural pruning. This also occurs later in adolescence, which is part of why people are constantly lecturing teenagers about being careful with their brain chemistry, and also about taking advantage of this neuroplasticity, because after adolescence and early adulthood, you essentially lose the ability to form these new connections, and your neuroplasticity goes way down because you've pruned so many of these connecting points. This pruning process occurs in two ways. You can have a set number of neurons. These little blue dots represent neurons, and then you can get rid of all of the different connections and only make a certain number of connections. This increases the efficiency of your brain activity. Or, let's say you have too many neurons in the first place, 3 over here on this side, 4 over here, you can get rid of actual neurons, and again, this cuts down the number of connecting points and allows you to access information much faster and makes brain activity more efficient. You might lose the ability to learn a new language fluently. However, you'll be much more efficient in the language that you learned when you were a small child. And so after again, 6 years old or so, you've established most of these neural connections you're going to make, and you just start to kind of cut them away to make this process more efficient. This is where I'm going to leave the lecture for today. We're going to keep these pretty short, just 15-minute segments. As we move on, we'll add more detail to these kind of foundational concepts that we covered today. I recommend you read the sections A.1 or 6.5 if you feel like you didn't quite get enough from this, or you want more detail, or you missed something. And please be sure to look at Teams for your assignment for the week. And that's it. I'll be signing off. Have a good one.